This Week in Virology, the podcast about viruses, the kind that make you sick.
Welcome to Futures in Biotech. Today, we're going to do an experiment. I guess as scientists, we are always compelled to, try to do something different. And this is going to be what I call a sci mashup. So we're going to do a podcast within a podcast, and we'll see if they coexist. And we think the audiences are fairly similar in the feedback that we're getting. And these guys do a podcast that I like to listen to. It's really fun. One, they're very experienced in their field. Two, they're great communicators. And three, fantastic public production quality on their shows. So I appreciate all three, and that's makes it really a great way to spend time when you're doing cell culture, when you're in the lab working. So without further ado, I'd like to introduce the cast of This Week in Virology. And first, let's introduce the producer, Vincent Racaniello. Hi, Vincent.
Hey, Marc. This is visit number 11 on FiB.
Really? Has it been 11?
It's amazing, isn't it? More than you.
Yeah, so Vincent's been co-hosting a lot of shows on Futures in Biotech because he's one of our subject matter experts and, a great podcaster. So we've relied on him for a lot of great interviewing. And so today he's kind of on both sides. He's being the guest and the host. Very meta. I'm just very confused. But hey, so the stories that we're going to talk about today, I'm going to do a little preface here, are very much related to some of the stories that are relevant to the shows that have been on recent topics on Futures of Biotech. So, Vincent, perhaps you could introduce your cast. It would be really great to.
Well, I'd be happy to. But first, let me say that this is This Week in Virology, episode number 125, recorded March 18th, 2011, and I'm Vincent Racaniello, as you heard. Sitting behind me here with his arms folded is Dickson Despommier. Hey, Dickson.
Hey, Vince.
Dickson's at verticalfarm.com. Good to see you.
Good to be seen.
Also joining us today from Western Massachusetts is Alan Dove.
Good to be here.
Hey, Alan. There he is.
Hey, and there I am on camera.
Yeah, with the wood on the ceiling. How are you?
Yes. I'm going to get rid of those beams at some point, I suppose. Doing okay. No more ice accumulating here, so that's a good sign.
Yeah.
Spring is on the way, and of course,
yes, so they say.
Where our third host is, our fourth host is Rich Condit. It's always spring. He's in North Central Florida. Hey, Rich.
Hi, fellas. How you doing?
We're good.
Doing great.
And that's our crew.
84 degrees, clear blue skies.
That's right. Rich also works for the Chamber of Commerce.
So today we have the usual TWiV. Marc, you want to just begin?
Sure, let's go for it.
All right, so we have two stories which we thought would be cool to talk about. The first one is called, it's a Science Express paper, it's called A Virophage at the Origin of Large DNA Transposons. And that title is a bit...
Yeah, and I just want to say that if people thought that the show was meta in the introduction, it's going to get a whole lot worse.
It is. So...
Before we do go, I'm just trying to piece together here who's who, and for the audience. Richard Condit, you did your PhD at Yale, right?
That's correct.
In the early 70s.
A long time ago.
With Joan Stites.
That's correct.
And so you were 2 labs down before I did my postdoc in 2002. So we crossed paths, but not in the 4th dimension, right?
So wait a minute. Were you in the basement of the Sterling Hall of Medicine? I think Joan had maybe moved by 2002.
Right. She moved. She was at the Boyer Center, so it's kind of meta.
So the Boyer Center didn't exist. It was a parking lot when I was there. But I was in the basement of the Sterling Hall of Medicine. I was actually her first graduate student.
Fantastic. Well, that's amazing.
The first one in, not the first one out. Okay, so it depends on.
That's right. But at least you got out.
Well, you probably should also know about. You should know about Alan Dove, who he is currently a science writer residing in Western Massachusetts, but he was my PhD student in the early 90s, right, Alan?
Yes, indeed, I was, and it was very nice of you to only specify the early 90s.
Oh, you were here in the late 90s as well?
Sorry,
actually
that's the 1990s, by the way.
Past yeah. But yeah, early 90s, that's right.
Wow. And Dickson, you're onto a really, really interesting project.
Nice.
And maybe you could just give us a brief summary on it, and
Sure.
and I'd like people to go take a listen to the episode where Vincent and Dickson came on. Vincent co-hosted, but Dickson was the actual guest, and he talked about this project. You could just briefly, and then we'll go on to the paper.
Sure. I mean, it's just about with urbanization the way it's occurring nowadays, and with the loss of land due to all kinds of reasons, climate change, overpopulation, natural disasters, etc. It occurred to our class some 11 years ago to look at other ways of growing food, and one of the ways that they picked up on was to grow it on the rooftops. And it didn't turn out to be very successful. At that point, I said, well, why don't we just move the idea inside and grow it inside buildings using whatever technologies were available then. But since then, much greater, more powerful technologies have arisen, particularly with regards to making LED lighting much more efficient and much cheaper. So that's the idea, is to make, vertical food, vertical farming inside the city, fresh, on-demand food whenever you want it. So that's basically the idea. It has gained traction, and indeed there are some real projects out there now, and one of them might actually come to fruition over the next year and a half in Milwaukee. So we're very proud of the fact that an idea from class actually escaped into the real world.
It's in Milwaukee.
Yeah. Will Allen, in charge of a group called Growing Power, has plans and has started to raise funds for establishing a 5-story vertical farm in the middle of Milwaukee, and got permission from the city to do it. So it looks like it's going to happen. And by the way, I have a virology context to put myself into also. I got my degree at the University of Notre Dame, working on germ-free animals, but every Saturday I sat around a table at which was Morris Pollard, who was, of course, the chairman of microbiology at that time, and a first-class virologist who did most of his work on psittacosis and rabies. And around that table were all of his students, and I was the only non-virologist. So by osmosis, I picked up a lot of stuff, which I'm now spewing out on TWiV as gee, I heard about this first back in, 1963 or 64, or, places like that. And so I've had some very nice contacts with virologists most of my adult life as a parasitologist, so I feel at home here.
Well, it's too bad we don't have the recordings of those conversations. So
Yeah.
those would've been-
Oh,
right.
you know what?
That's right.
They were fantastic because they sounded just like our show. Because Morris was a very conversant person and very casual, but he you had to have performed in the lab that week in order to have something to say. Otherwise, he'd just pass over you. And so everyone made sure to do their work during the week so they'd have something to say on Saturday.
They would have had to record those like on a wax cylinder or something.
That's right. Exactly. A cat whisker with a... Yeah, they didn't have the modern technologies that we have today, but it was a lot of fun and very informative.
Dickson was my initiator of TWiV 3 years ago now.
That's right. I was your straight man. So
I
to
said,
speak.
who can I get to do this with me? And Dickson came to mind. So he started it with me.
I've had so much fun, and I've learned a great deal just by sitting here.
There'll be a test next week.
Someone can take it for me, believe me.
Your listeners are getting, an Ivy League education here,
Yeah.
and get to participate in discussions not only at the level of the undergrad listening to the topics, but getting the insight in a more informal way. And if they can, reach out to you and like they do with emails, they can converse with you and participate in the discussion. So it's really amazing. So, but right before we go, I just wanted to show this picture, which is a picture of Dickson in front of a prototype vertical farm here. And if I was in Milwaukee and one of these was on Lake Michigan, I would really appreciate the fresh vegetables out of season. And I think every city could really benefit from incredible quality and great use of resources here. I think it'd be just fun.
Thanks for the plug, Mark.
All right. So sorry to interrupt, Vincent.
No problem. No problem.
It's really, really fun. You guys are great, so you should celebrate the interesting chemistry that you guys have.
I should say that Alan was the second to join us. I thought it would be good to have a science writer, and that's what Alan is, has a different perspective. Of course, he has a broad view of science as well as virology, so he has a different voice that he brings to the equation. And then a bit later, we brought in Rich Condit, who he got it from day one. He was a guest, and he really got it, and I thought this is a guy we have to have on the show.
Absolutely.
So that's where we are at 4, and occasionally we have some guests come in, but it's mainly the 4 of us.
It's awesome. So I recommend that people who listen to Futures in Biotech, they send their RSS feed, RSS, what do you call it, aggregators or whatever. iTunes. Just subscribe. Subscribe to TWiV. I do. It's a lot of fun. And it was somewhat
so we should give them an actual episode now.
I think so. Yes, this would be good.
We could do that. So let's go to that first paper.
Viruses that parasitize viruses that parasitize other microorganisms, right?
It's getting meta, Alan.
I think this is appropriate since it's a podcast within a podcast.
That's right.
Yes, good point.
Podcast phage or something like that, right?
So this is a virus of a virus. It is been given the name virophage, and what it is, it begins with a very large virus. So those of you who have listened to TWiV remember the Mimi viruses, which are the largest known viruses, have a genome of double-stranded DNA over 1 million base pairs in length, with over 1,000 open reading frames or possible protein coding regions. And these are typically viruses that infect amoeba. In this paper, we're dealing with a similar virus. It's called the Cafeteria renbergensis virus. So Cafeteria renbergensis is a flagellated phagotropic protozoan. And it's called cafeteria because it eats all kinds of stuff in the water. And they found... Sorry?
Good and bad? Cafeteria.
Well, they eat
my cafeteria.
Mostly bacteria, I believe, which is what you get in cafeterias.
That's right. Another name they could have picked is forget it. Smorgasborgensis.
That's right.
Right.
So this Cafeteria renbergensis virus turns out to have a very small virus associated with it. So they look at infected cells, and they can see these tiny particles, which they call mavirus, M-A-V-I-R-U-S, which stands for maverick virus, and we'll explain why that is in a moment. But this maverick virus is much smaller in size, and its genome is also smaller. And it will only replicate in cells that are already infected with the Cafeteria renbergensis virus. Not only it needs the Cafeteria virus to replicate, it replicates within the actual places in the cell where the Cafeteria virus is growing. These are called factories. And this little virus, mavirus, replicates in those factories. And on top of everything, it inhibits the replication of the big virus. So it's an actual parasite. It requires the large virus, and it affects it negatively. Would you call that a parasite, Dickson?
I would. But I have to know something else, though, Vince.
We should point out that these, when you say the large virus, these are the Cafeteria virus is like the Mimi virus. It's one of these ginormous viruses. It's not just a little on the heavy side.
Right. It's a huge
750,000 base pairs, right?
Right. So a bit smaller than the Mimi, which is 1.2 million, but a respectable size.
Right.
Yes, these
Still
are huge.
three times or so larger than what we have in the past thought of as the largest DNA-containing viruses. And just for the listeners, I have a pretty common thumbnail calculation that I do where I figure that 1 kilobase of DNA is enough to encode about one gene. So 750 kilobases is about 750 genes. And the Ma virus is 20 kilobases. So that's about 20 genes. A lot smaller, kind of a medium to small size virus as they go.
All right.
Do most viruses code double genes, by the way? Like where the DNA can, on one direction gives one gene and that same DNA in a different direction gives another gene?
Yeah, that happens. You have coding regions on both strands, sure. Especially in the smaller...
Not very frequently. Oh, well, coding genes on both strands, but not necessarily completely overlapping.
That's kind of like Scrabble. If somebody's trying to imagine this, DNA can work as Scrabble, where you have one word going one way and then another word starting halfway up going the other way. Or
Sure. When Vincent and I had this conversation earlier this week, I asked him what the difference between this situation was compared to what I already knew about helper viruses, like SV40 and things of that sort.
Well, that's a good question. So there are some known viruses that require a helper,
right,
in which they can replicate. And a good example are the so-called adeno-associated viruses. They are small DNA viruses. They need to be in a cell co-infected with adenovirus.
Right.
There was also hepatitis delta, which requires hepatitis B to provide a capsid. So that's a helper virus. Now, why this is different is because this little May virus actually inhibits
that's
the big one.
Exactly. So that's a non-helper.
Right.
Or it's a cure for the Does it cure the infection of the big virus?
I think it decreases
it doesn't cure it, but it inhibits the replication of it significantly.
And the other question is, does the Ma virus replicate on its own, or does it actually need the larger virus?
Right. It does not.
No, it needs the big guy.
So from one standpoint, there is a helper virus. The bigger virus is the helper virus for the little one. And the little one actually inhibits the bigger one from reproducing. That's kind of bizarre.
Right. So the way to look at it is that the little virus is parasitizing the big virus.
Yeah.
In true sense, it is infecting that virus and inhibiting its ability to live.
Sure. So what is the
you raise a good point though, Dickson, because I've always thought I've thought with Sputnik, which is the virophage of virophage.
Yes.
The word virophage was kind of stretching it. It seemed to be more like a satellite. I think the differences are subtle. In this particular case, a couple of things really impressed me. One is these electron micrographs that show the Ma virus assembling actually inside the factories of the co-virus. All right, so that looks a little more like a real parasitism. Plus, they have this this analysis where they've looked at the transcriptional signals in the Ma virus. Now, this is bioinformatic. It's a little speculative, but the transcriptional promoter sequences in the Ma virus, or putative transcriptional promoter sequences, look the same as in the co-virus, the big guy, as if the Ma virus is using a transcriptional machinery from the co-virus, and so that looks a little more like a real molecular parasitism.
The other thing that's interesting here is that you mentioned before, Dickson, is this good for the small virus, or is it good for the host? Because the small virus, and in fact, they say here, eukaryotes that are susceptible to infection with these giant viruses will gain an advantage if they can associate themselves with these virophages, because they will protect them.
Sure.
So maybe that explains the evolution of these small virophages, if you will. I don't like that name. What do you think, guys? Is a virophage not a good name?
I've never been crazy about it. This the justification in this case seems stronger than in the case of Sputnik. It seems to me. I've never been crazy about it. But you know what? What the heck? We're stuck with it.
Well, my problem with virophage is a phage is a virus. So we've taken a term that means virus, and we're using it to describe a subset of viruses in another context. I think it's a little confusing. I don't know. I might call these something like epiviruses, or I'm kind of at a loss for a really good term for it. But virophage seems a little off.
It's a competition, though. It sounds like, for resources that
sure.
If you're going to put so much into the small virus, you can't get as much big virus, and it's very does the small virus ever integrate into the genome of the big virus?
They actually looked, and they didn't find any evidence for that. Which doesn't say it doesn't exist.
Vincent.
Because
here we're you guys are your TWiV audience will be like the cafeteria virus infects the
right
the Maverick virus and they'll get it. So I want to not that my audience my dad's listening, so I want to explain it to him. So you got this cafeteria virus, which is a giant virus, like 730 kilo. That's 700,000 bases, giant genome,
right?
And it parasitizes. Oh no, wait. the cafeteria is only 20 genes or 20 kb.
No, cafeteria is the big one.
The cafeteria virus is the big one, the 700,000 bases.
Wait a minute, I thought the cafeteria was the host.
The cafeteria is the host.
Yeah, that's the flagellate. That's even bigger yet.
But the virus is also called cafeteria virus.
Confusing.
Right.
That's confusing.
Oh, okay. Oh, yes. So the virus, the small one, the virus, the 20 kilobase genome, 20,000 pieces of DNA or bases.
Yeah.
In the mavirus, infects the cafeteria, which is the large one. So you got this little small virus goes in and infects a larger virus,
right?
The cafeteria
which is
virus.
Infecting something even bigger yet.
Which is infecting
at least it infects the cell containing the larger virus. A lot of debate here has been whether you can really call it infecting the virus, and that's where we're having a debate about whether to call it a virophage, which implies that it actually infects the virus, as opposed to just a satellite where it's using the bigger virus as a helper for its replication.
So
and also just to beat a horse that we hit frequently here on TWiV. It depends. I mean, can something be infected if it's not living?
Right.
Oh, no.
It sounds like two competing viruses for the same set of resources to me. That's what it sounds like. You've got a viral factory going on, which has no complete virus particles whatsoever, and you've got a choice. You've got so much resource. This host cell has only got so much to give, and these viruses are competing for that resource. And it apparently, when the little one is present, the bigger one doesn't get as much as it usually gets.
Right, the factory is normally building Mack trucks, and somebody's using the equipment to build bicycles, and that takes away from the resources that they would.
Something like that. That's right. Yeah.
So one thing we haven't mentioned yet that's an important consequence of this, probably as an indirect consequence of inhibiting the replication of a large virus, is that the cells live longer too.
The host cell. There you go.
So there is an evolutionary advantage for the host cell to pick up and harbor this little virus, because the little virus inhibits the growth of the big virus and the cell lives longer.
And this is a marine flagellate, correct?
A marine flagellate.
Right.
All right. So lots of stuff going on out there in the natural world.
Lots of stuff going on. Well, this guy, the senior author on this paper, Curtis Suttle, he's responsible for he's done a lot of work on characterizing viruses, marine viruses. Nice. He's responsible for some of these numbers about there being 10 to the 11th phage particles per milliliter of seawater.
10 to the 6th.
A ginormous
10
number.
10 to 6. Okay.
Fine. A lot of stuff out there. Yeah, exactly. You know what? Someone has likened this to the true carbon sink for the ocean.
Sure.
I mean, that may be where all the carbon is actually going.
Yeah, the lysis of bacteria and other
can't see it.
organisms by viruses in the ocean. Yeah. Absolutely.
Yep.
So the other story here, which is very interesting, is that this virus has genes which are also present in jumping DNAs called transposons.
Uh-huh.
All right. Now, I thought, Mark, your audience would like this part. There are a couple of different kinds of transposons. There are transposons related to retroviruses. That retroviruses encode an enzyme that can copy their RNA genome into DNA, and they also have an enzyme that can integrate that DNA into the host. So that one kind of transposon employs that mechanism. And then there's another kind of transposon which is just made of DNA, and it can jump around the genome. And here's the part I think your audience will like. There are two mechanisms for movement of these DNA transposons. There's copy paste and cut and paste. Right?
Sure. Well, that makes sense, right? You can either copy it and double it.
That's it.
Exactly.
Or you can snip it out and replace.
So the cool thing about this Ma virus, and the reason they call it Maverick is because Maverick is a name for a DNA transposon, is it has genes that are also found in these transposons. So it may be some kind of an ancestor of transposons. And that's
so they do some analysis of this evolutionary relationship between these two. It's not just a passing resemblance. It's actually a pretty strong homology between these two that suggests that either the Ma virus came from the transposable element or the transposable element came from the Ma virus. And they argue, I think pretty persuasively, that that it's the Ma virus was the ancestor that gave rise to the transposable element. So this is, we've got these transposable elements hopping around, and there's been a debate for a very long time. Well, did viruses come from transposable elements, or did transposable elements come from viruses? And I think this is a case, at least one case, where the evidence would point toward the transposon coming from the virus. So you had a Ma virus ancestor, they call it, infecting some cell in the primordial ocean. And it ended up transferring its DNA into the host DNA and integrating and becoming a transposable element in that DNA.
Could it be both? I mean, could, biologically speaking, there be both? I mean, the fact that we can think about it plausibly would suggest that chances are there are both pieces of DNA that jumped out of a genome and became a virus versus the other possibility of the DNA, the virus infecting and becoming a permanent piece of DNA inside the genome that then became
it's not necessarily a one-way trip, I would think, because once you're integrated as a transposon, I mean, it's not a huge leap to go from there to being a virus again. In this particular case, it does look like it probably went from virus to transposon, but yeah, I don't know. What do you guys think?
Well, this brings up some subtleties about the genetic content of both of these things, because one of the things I notice is that the genes that are homologous between the Ma virus and the transposon include a capsid protein and an integrase, right? Both of these things have both of those things. Now, if you got a capsid protein, that implies that you would encapsulate the DNA at some point and you would be a virus. So it doesn't necessarily make any sense that a transposon would have a capsid gene. Conversely, if you're a virus that doesn't integrate, it doesn't make any sense to me to have an integrase. Right. So it implies that both of these things genetically are sort of positioned, if you like, between transposon and virus, or capable of doing both, or have done one or the other recently. So it's an interesting situation. The other gene that interests me, this is a little more subtle maybe, is that it's a polymerase that copies DNA using a terminal protein. This is a mechanism that's used for a couple of bacteriophage and an adenovirus, where the DNA is linear and has a terminal protein and uses a special polymerase to do this. But the Ma virus genome, according to these guys, is circular. So that doesn't make any sense to me. So there's some funny business going on with the genetic content that has implications for the evolution of these viruses.
It's also worth pointing out that the name of this virus was named after Mariner. Mariner is a kind of transposon, and we have quite a few of these Mariner transposons in us. We have about 14,000 copies in our genome, which is about 2.5 million base pairs. And I don't know if any of you have ever heard the science fiction novel called The Mariner Project, but that was sort of inspired by this Mariner transposon.
By the way, transposon is a jumping gene to the general audience here. A transposon is a small segment of DNA that acts somewhat like a virus, I suppose, in that it can jump from out of the cell and throw itself into another cell and integrate into that genome and pass genetic information on.
Yeah.
So should we talk again about whether viruses are alive? Okay, because it always raises if viruses are alive, then you have to ask, are transposons alive?
Well, how many more genes does this large virus need before it becomes classified as another kind of organism?
Good question. People have said this is sort of bridging now the gap between viruses and
chlamydia
and
would
cells.
be the next group up.
Are there bigger viruses? What is the actual limit to the size of a virus?
Or when you pare down, I know they did this experiment with E. coli. They just cut out all the non-essential genes, and what did they end up with? Like 365 genes in a culture plate supplying almost all the nutrients. You could still get the bacteria to look like a bacteria. That has something like 3,200 genes.
Rich, an answer
smaller than this. Yeah, smaller than this virus.
Exactly.
The idea of a virus replicating in a virus is wrong because the virus doesn't have any resources to provide the other. What can happen, as we know, in this case, the virus replicates in the cell, and then the other virus parasitizes the replication site.
Right.
Right. If you define the virus as only being the genetic material in the capsid floating around free in the world, then that's true. A virus can't replicate in a virus. But if you define the virus as its life cycle, then a virus can certainly replicate within the life cycle of another virus and use its machinery and parasitize it.
Doesn't that happen a lot, though, with viruses like influenza, that sometimes you can get two different strains into the same cell, and they co-mix and co-exchange genomic material. So that's not an unusual situation. But in this case, though, the little virus is inhibiting the growth of the big virus, and that's probably the basis for calling it a parasitism rather than something else. But actually, it might just be resource reallocation.
It's absolutely crazy. There's so many levels here. Transposon, small virus infecting big virus infecting host cell.
And also, if you think about the possible origin of this, they speculate a little bit about this in the paper, and the upshot of it, as I understand it, is that probably at some point in the distant past, some May virus ancestor infected or managed to evolve within the context of an infected cafeteria ancestor in the plankton. And it provided a selective advantage because it was this, the enemy of my enemy is my friend. So the virus of my virus is my immune system in a certain sense. And if you're a little one-celled organism and you're swimming around and you've got this big virus infecting you, and you can harbor this little virus that nips at the heels of the big virus, then that's an antiviral. It's an antiviral defense system. So to some extent, there's a selective pressure to keep that little virus around, and integrating it into your genome would be a way of doing that.
Mm-hmm. Yeah.
So this could also be the origin of an immune system.
Interesting. And then the transposon becomes a series of mutated forms of the same antibody, so to speak.
Let me ask I've got just two basic questions. The first one is, I'm trying to visualize here a host cell with a baby virus or a small, the maverick virus, and then the big virus. Now they've lost their capsids, and it's just DNA floating in the cytoplasm, or is it in the nucleus, and they're functionally interacting together, or do they actually fuse and interact and become one?
There's the big virus is a cytoplasmic virus, as I understand it, and it sets up replication factories in the cytoplasm. And in the supplementary material for this paper, there's a beautiful electron micrograph that shows a virus factory, and it's surrounded by some assembled big viruses, the cafeteria virus, and inside the factory you can see assembling the little virus. So they're happening. The little virus is actually apparently replicating inside the factory that's normally established by the big virus. And the factory presumably contains the big virus DNA. So the DNAs of the two viruses, it looks to me as if they're mixed together in this factory that's normally established by the cafeteria virus. Wow.
The second
is right.
The small virus, this maverick virus, which has a genome that looks very much like the genome or the DNA sequence of a jumping gene. This mobile DNA element that probably is involved in evolution. Now the last show that we did on Futures and Biotech, we had Susan Lindquist on talking about how a heat shock protein, which is a protein that stabilizes other proteins during stress, could respond to the environment. And when the environment has very stressful conditions like heat, it would titer out this heat shock protein away from its target proteins, which it normally acts on, which are signaling molecules and information molecules that then go and And by titering it away from those very important signaling dip switches, information dip switches in the cell, you would spawn a phenotypic change. And she was suggesting that this is a major link between the environment and the environment having an active role in spawning evolution. I asked her, is HSP90 the only or the most important element in spawning evolution? My question to you guys is this maverick virus, the small little virus which can then turn into a jumping DNA, a piece of jumping DNA, or even just the jumping DNA, jumping gene, are those important elements in our evolution?
Absolutely.
Oh, no question.
Absolutely.
Not just transposons, but all viruses are known to move DNAs around from organism to organism. So they're huge role. They play huge roles in evolution.
This is a lateral way of changing your genome rather than having to go through reproduction.
So do you want to get the flu?
Well,
no,
Marc, you can get other viruses that don't make you sick, and they can do good things. And I think what's happening in nature and animals that we don't know much about is that there's a lot of movement of genes by viruses that are not harmful in any way. And we're just starting to learn about this.
Right, and we certainly know about this from bacteria with bacteriophages, and you have phages that can confer abilities to survive in other environments or antibiotic resistances, and it's useful genes being transferred around by viruses.
Well,
and we talk about this on TWiV all the time, but the Futures and Biotech audience may not be as familiar with this. The human genome is a very large percentage of it is carcasses of old viruses. Right.
Yeah, 40%.
Yeah. I think we discussed it with Marc Gerstein, who's kind of a bioinformatician that looks at the genome as an archive, right? A historical record.
Yes. It is, sure.
Sure it is.
It's a fossil record for sure.
That's right.
Hey, Marc, can we move on to our second paper? Because I think this is pretty interesting, too. This is called Unintended Spread of a Biosafety Level 2 Recombinant Retrovirus. And Alan...
Oh my God, we're all going to die!
So, Alan,
I'm feeling a little peaked myself.
But we can qualify that.
Alan, you discovered this. Why don't you summarize it for us?
Yeah, so these folks, this paper actually came out in 2009, but apparently nobody noticed it. And I was doing a search on something else, and I stumbled across it, and I said, "Wow, this is really, really interesting." Especially in light of some of the other discussions we've been having on TWiV about contamination or potential for contamination of cell lines with various retroviruses. And so these folks, a German group. They were working with this cell line, a very common cell line, 293T cells, which is I think a human embryonic kidney cell line, and they had these vesicles that were forming in the cell culture, and they wanted to look at them and see, well, what's going on with these vesicles the cells are forming. So they did an electron micrograph, and they saw that the vesicles were budding off things that looked very much like, there's the picture, that looked very much like retroviral particles, which is not quite what they'd expected because this wasn't supposed to be an infected cell line. So they went and looked, and they did a sort of an open-ended RNA PCR, RT-PCR, and also regular PCR to see what the genomes were like in there. And ultimately what they pull out is two different retroviruses. One is what is it? It's a squirrel monkey retrovirus, right? And the other, so that's interesting. You've got your cultures contaminated with the squirrel monkey retrovirus that I don't think they'd worked with. But the other is a real bizarre one. It's a murine leukemia virus-like virus. But it's not XMRV, and it's not a normal wild occurring murine leukemia virus. They did a database search, and what popped up was a recombinant retrovirus that was created in the mid-1980s sometime. So this is a virology lab was working on retroviruses, created this recombinant, made a plasmid of it, and these folks in this German lab have never worked with this plasmid. But it has near 100% identity with the retrovirus that's budding out of their 293 T cells. So somehow, probably by swapping cell lines with other labs and working with collaborators and who knows what other mechanism, they got this recombinant retrovirus that was made in some other lab 30 years ago, and that just is now inside their cell culture. And they do some subsequent experiments and determine that both of these viruses are fairly promiscuous. They'll replicate in a bunch of different cell types. And they also went to their freezer and looked at other samples they had of their cell lines, and they found they had a whole bunch of them that were contaminated with this sort of thing. Sure. So it's a systemic problem. And to me, the really surprising bit was that one of these viruses came from a lab.
That's not good news, is it?
Well, that one of them came from a lab and was then transferred to other labs that weren't working on it unknowingly. Right. And the reason this is interesting for a recombinant retrovirus is because these are supposed to be handled under what's called biosafety level 2 or BL2 or P2 conditions, which is not, that's not the whole bubble suit or what have you that you see in the movies, but it's some degree of precaution that you take to keep the virus from getting out into the world. So you're supposed to bleach things before you throw them down the sink and autoclave your, virus when you're done with it, and store things separately and not cross-contaminate. But clearly, that is not sufficient to keep this from getting sent around. And people who are working with murine-like retroviruses or discovering murine-like retroviruses are susceptible to exactly this sort of thing. And there's no telling how widespread it is.
I think
people exchange cell lines all the time. I've sent cells we work with, I've sent to a number of different laboratories over the years. So all you would have to do is get a cell line that's contaminated from another laboratory, and then there's the potential for contamination to other lines in your own laboratory, and then you send it somewhere else, and somebody else gets contaminated with the same thing. So it's once you know that this is a possibility, despite BSL-2 containment, it's not a surprise. It's not a surprise.
I think many labs have contamination that they're not aware of, and this paper underscores how easy it is. It's pervasive. And I think, Marc, if you checked many of the cell lines in your company there, you might find some viruses as well. I mean, the point is that these viruses are not harmful to the cells. So if you look at your cells in a microscope, they look completely normal. But yet the virus is there. The cell has evolved to deal with the virus in its own way. So you can't tell. In fact, Marc, they used mass spectrometry to look at the supernatants of these cells to identify the virus in there. Can you imagine? They took the they did total. You don't need it, but that's what they used to figure out what was in there.
Right, and the reason they had to do that was because there's no telling what the cell line might be contaminated with. They just stumbled on this because they happened to, I guess, have an electron microscope sitting around and the ability to use it. And so they looked and they saw these retrovirus-like particles, which means it pretty much means that there are going to be other retroviruses out there contaminating other cell lines, and we don't know what, and we don't know how widespread this is, but I would be willing to bet that this is going to be a nearly ubiquitous problem.
Wow.
You can control. So the whole point of, for example, we use cell lines, we use Chinese hamster ovary cells, and we express a gene, we overexpress it so that we're well above the endogenous levels of protein. And we look at the functional response, the physiological response of these cells as they're made. And we have a control cell line expressing a different protein without that function. And now we're eliminating all cellular effects and just looking at the numbers that we can collect that differ between the functional protein and the non-functional protein. So I guess with controls, you can really not worry too much about these viral infections of your cell lines. But I suppose if you're trying to study something that's an endogenous level protein, something that's native to the protein to the cell, I mean, then you
I think if you're doing basic cell biology, this is probably not a big concern because, as you say, you can control for the other factors simply by using the same cell line in your experiment and your controls. But where this comes up, and where the reason it really came up in my literature search was we've been talking on TWiV about XMRV, which was discovered in 2006, and we are now getting increasing reports that it may have been a lab accident in itself. And this paper underscores just how something like that could spread. And it also underscores the caution that you need if you're going and looking for viruses. So this is not something that undermines all cell culture experiments by any means, and I don't even think it undermines virology experiments where you're using a lytic virus, for example. But what it shows is that if you go and you look for retroviruses in a cell line, or you look for other viruses that could be in there without killing the cells, you may very well find them, and it may very well be meaningless.
What if, let me pose this question, if you have a bioreactor and you've got HeLa cells growing and you're using it to amplify a strain of influenza that's going to be used as a vaccine, do we currently have methods to verify that we're not making an extra vaccine at the same time?
Well, the answer, Mark, is that you wouldn't use HeLa cells because it's a transformed line, and we can't do that. So you have to use cell lines that are qualified for use in growing vaccines. The answer is yes, we look by PCR to make sure there aren't other viruses in there. But last year we talked on TWiV of an example of an investigator who bought 10 different human viral vaccines, and he deep sequenced them and found other viruses, including a retrovirus, in some of those human vaccines. A few weeks ago, we talked about pet vaccines that are contaminated with retroviruses. So we still are not really 100% at detecting all the contamination, at least during the production.
Having said that, the safety testing on vaccines would certainly catch anything, and the aftermarket monitoring on them would certainly pick up anything if these viruses actually had an effect. But it is, it is potentially a problem for people at the research stage, I think.
There was another report that I think we discussed here I just looked it up, from 2009, where Genzyme had a problem, where their bioreactors got contaminated with a virus.
That's right.
And that shut them down for a while. So the good news is that people do check for this stuff. And the bad news is that it's a problem now and then. I must say I had the cynical thought as I read this paper that if I could have a publication for every time I contaminated my cells, I would have a really long CV.
Yeah, exactly.
Well, some of the early conversations that we had around the table back in 64, 65, were just about that. What constitutes a germ-free animal? You can check for fungi and bacteria, and of course protozoans and helminths are out of the question too, except for one. Well, I won't even talk about that one, but you can prevent that simply by cesareanly sectioning the pregnant female and carrying the embryos into the, or pups, I should say, into the incubator and making sure that they're sterile. But transmitting viruses vertically in the genome and in the cytoplasm was the biggest worry that they had. And I guess even at the organism level, it's still a big problem, and one which, if you don't look for it and it's there, it could alter. Remember what happened when everyone found Sendai virus in their mouse colonies and found out that Sendai virus, of course, it comes from that little town that just got creamed, unfortunately, which reminded me of it. It inhibits the immune system. So all the experiments that all these people were carrying out with these mice that they thought were pristine were actually under some immunosuppression induced by their viral infections, and they had to redo all those experiments, and it's very expensive.
Right. Yeah,
I think we should. Go ahead, Reggie.
No, I was recalling. Dickson reminded me that there was a talk that I recall from a couple of years ago from Skip Virgin's lab about discovering a mouse virus that they weren't aware of before. That, as I could vaguely recall, influence the immune response of the mouse to another infection. Okay, so there's potentially all kinds of stuff like this going on that we're not aware of that we'll find out bit by bit.
Yeah, I remember the Jackson Labs went crazy trying to get rid of it.
Yeah, and the ones that have been found have often been found in the context of somebody getting a vaccine or biological product ready for market, where they are going to go and do this due diligence and search for everything under the sun, and then they'll find, oh, hey, you've got this virus in there. But that has not been something that people did with their research samples because nobody thought it mattered. And to me, what this paper really brought home was actually it can matter depending on what you're looking at.
Sure.
Actually, we've talked on TWiV several times in the past about getting somebody on who knew something about how products were screened for adventitious agents. And there's just recently been a meeting in Washington about this following on the contamination of the rotavirus vaccine with porcine circovirus 1. It would really be interesting to get somebody on the show who knew something about this and could tell us how things are usually screened and what the plans are for the future. It actually might be a good topic for Futures in Biotech.
Yeah, I think that's a good idea. We can identify someone who does that. But I would like to leave this with one final thought, and that is we're not trying to be alarmist. All the vaccines for humans are well tested, and there are no adverse effects that could be attributed to these contaminants. So I don't want people to think that these are making vaccines unsafe in any way. We do find these adventitious agents from time to time, and we suspect that they are inconsequential because the clinical trials of the vaccine show that they're safe.
And they could even help.
They might even help. Who knows? Yeah.
And the technology is improving all the time. So, the good news is that we are discovering these things, and the technology is getting better all the time and will help us make the safest products possible.
Here.
Well, and in fact, a major reason that people had not noticed this in the past was that these things had not been causing clinical problems. I mean, if you look at the safety profile for particularly vaccines, it's astonishing how safe these things are and how closely that's tracked.
Sure.
But because it's not been tracked in a research context, nobody noticed these non-clinically relevant viruses that are just being transferred because when you exchange cells with a colleague, they're also exchanging cells with everyone they've ever exchanged cells with.
Sure. The biggest worry in the real world here, I think, is organ transplantation. And the fear that if you could overcome the heterologous graft versus host response or host versus graft response with some trick so that you could use pig organs or some other animal organs, monkey organs, that you'd still have to worry deeply about the hidden virus infections that might still be there.
Yeah.
Mm-hmm. But, we shouldn't also spend your whole life scared. Anybody who has a kid in kindergarten will realize that the schmutz that's all over the place is just
that's right.
And any parent will know that every 3 months they're going to get a weird cold, one that gives a headache, one that gives a sore throat, one that gives an upper respiratory tract.
And to their parents.
Yeah, not just the kids. Oh, sometimes the kids, 24 hours later, they're like, boom, back. It's the parents.
That's right.
Mark, I suspect that these infections at a young age are actually important for educating your immune system. And there have been a number of studies, as Rich referred to earlier, which show that some infections can confer protection against other microbes. So we still have a lot to learn, but not every infection is bad. I'm sure of that.
Right, and of course you don't want to get the infections that can kill you. But the ones that are subclinical, sure, they're showing your immune system something.
Do you guys all, one thing that's really important is to ask the pros what they do. And you guys are pros with respect to viruses. Do you guys get the influenza vaccine when the season hits? Do you go out and get your shot?
Yep.
Yes, absolutely.
Yep.
Everyone?
Absolutely.
Yep.
Yes.
Well, so do I. I just want to say to the audience, yes, go get your influenza vaccine. You want to save your community. It's not just for you, but you're also preventing the spread to others that may not be as resilient. Elderly folk, people that are immunosuppressed because they've received a transplant, kidney transplant, cornea transplant, anything, they're immunosuppressed. So you want to, not just for your sake, you hear a lot of people say, "Oh, I don't need to get it. I'll survive it. It's just a week, and I'll get over it." But you're passing it on, and you want to be good to your family and good to your friends.
I just gave a flu lecture to my virology class today, and pointed out to them that the average yearly excess mortality from influenza in this country is 40,000 people a year. That's a big number. Right. Flu is not just another cold.
Nope.
And just to expand on this, it's not just that I go out and get the flu shot every year. My daughter's fully vaccinated. My wife is on the board with all this too, and we absolutely take this seriously.
Speaking of vaccines, if we could move on to some emails, Marc. The first one has to do with vaccines. So on TWiV, we have a great audience that interacts with us via sending questions by email. And I thought it would be fun to show the Futures in Biotech audience some of these. We have so many that we can't possibly read them all. It takes us months to catch up. But we have some lined up today, and I thought we would go through some of them. The first one is from Todd, who writes, "I'm a computer programmer who listens to podcasts on my long commute. The highest science education that I've had was college chemistry. As an electrical engineer, the science classes we took tended to be not biology-oriented, so while some of it is over my head, to say that I find biotech and molecular biology fascinating is an understatement. Virus-related question. I also have a picture of me with the mumps at around 4 years old, 1974. When did the measles, mumps, rubella vaccine come out? Was I one of the last in my generation to have it? I've never met anybody my age who's had the mumps, only older than me. P.S. Lately, I've been listening to the FiB, TWiV, and TWiP podcasts to the point where I'm neglecting my techie podcasts. You guys are causing me to lose some of my geek creds."
Uh-oh.
So I thought...
this is uber geek stuff. This is uber geek.
Uber geek, that's right.
So Rich and Alan, you have answers to this question.
Yeah, so the first mumps vaccine was made in 1950 and used. It was a killed vaccine. That means they grow up wild-type virus and they inactivate it with formalin or something like that, so it doesn't actually replicate in you. And that was used between 1950 and 1978, but it didn't give very long-lasting immunity. The first live attenuated vaccine was developed in 1967, so that overlapped the use of the killed vaccine, and that was a better immunogen. And Alan, you came up with the MMR,
right? So then 1971, the MMR combination vaccine came out, and that was, of course, like most of the vaccines from that period, that was a Maurice Hilleman product. So that came out in 71, and that's what I would expect he would have gotten.
Right. If he was, let me see, what did he say? He got the mumps when he was 70. So he was born in 1970.
Right.
So he would have gotten, I suppose he could have gotten the inactivated vaccine, but more likely that he got the MMR vaccine or the live attenuated one. These vaccines are not 100%. There's, they're in the 90% or 90% plus. And that was shortly after the vaccines first came into use. So I don't know how suppressed wild type mumps would have been at that point, whether it was really down to baseline or not. But it's not impossible to get the vaccine and also get the disease. It's
right, particularly
rare.
if you're in an area where a lot of other people have not gotten the vaccine,
right.
And then there won't be the herd immunity, which is one of the reasons that everybody needs to get the vaccine, is so that you don't, so you don't kill other people with it.
All right, our next email is from Peter, who writes, "I really enjoy TWiV and especially TWiP. The Socratic method to teaching parasitism works for me. I am an aerospace engineer, but I now work in IT. I'm an engineer at heart, but I find parasitology fascinating, and viruses are like little machines, so also fascinating to an engineer. In your recent TWiP discussion of Giardia, you noted that the UV pen is ineffective against cysts because of their low metabolic rate, or more specifically, I think it was because their DNA was inactive. I didn't know that was a requirement for UV to work. So I wonder, is UV light effective in killing viruses in water? Mm-hmm. What did you say about Giardia, Dickson? Do you remember?
Good question.
You don't remember?
We'll have to go revisit that episode to find out exactly what I said. But if an organism isn't dividing, then there are certain precautions that don't work versus ones that do work. So maybe that's what we were discussing.
So UV will cross-link DNA.
Sure, it makes thymine dimers out of DNA.
So if the DNA is not replicating, it will not have a lethal effect.
That's correct.
But as soon as the DNA begins to multiply, then it will be dead in the water.
Exactly.
Okay, so maybe that's what you were saying.
Maybe that's what we were
So it won't kill a cyst because a cyst is a non-replicating form.
Here.
Right?
That's correct.
But as soon as the cyst begins to germinate, would that be the right word?
Unless, of course, its repair mechanism comes along and fixes the dimer. Right.
Now, as far as water goes, UV doesn't go very far in water.
It does not.
So when we inactivate viruses by UV, we make sure they're in a very thin layer.
We pasteurize
water.
Water. Well, that's basically the point. You make a very thin layer.
Yes, so the UV can penetrate. Otherwise, exactly right. It won't go into a tube, for example, of an inch in diameter.
You got it.
He says, I seem to recall that radiation is an effective treatment for cancer because cancer cells divide so rapidly. Is that the same effect?
A little bit different idea.
Well, you are killing cells, and there are more cancer cells because they're multiplying quicker.
Sure.
So you have a higher burden.
And the cancer cells usually have defects in their DNA repair mechanism, so they're more susceptible to radiation than healthy cells.
That's right.
That's right.
Anything else to add to that, gentlemen? All right. Hey, Marc, are you there?
I'm right here.
How we doing on time, Marc?
We're fine.
We're okay?
Yeah, we're going to make it.
Should we do it?
We're going to make it. That's great.
Should we do another email? Is that okay?
Absolutely. Go for it. Yeah.
All right. We have one from Aaron. Dear TWiV, I know my last set of questions about endogenous retroviruses was long, but I'm still holding out hope you can answer some of them. I've sent similar questions to several virologists and haven't received any answers. Well, Aaron, this is the place to go. In fact, we answered these a couple of episodes ago. I've heard you guys talk about viruses evolving to become less virulent. Could you explain this to me? Are we really saying that the immune system of their hosts have evolved to resist the viruses rather than the viruses changing something to become less virulent? My understanding of viruses is that they harm the host merely by reproducing and using up the cell's systems. I understand that very few produce proteins that are actually toxic to the cell. Since viruses consist of only a handful of genes that are dedicated to the production of new viruses, what genes can be mutated to decrease the virulence of a virus? It seems that if one of the viral genes are seriously mutated, the virus will be unable to produce new viable viruses. Will that mean the virus is less virulent? It also means that the virus will cease to exist. On the other hand, a virus host is a complex organism with many genes. A host can experience a mutation in an immunity gene that allows it to resist an infection without changing anything else in the host. A virus doesn't have this luxury. Any mutation will either cripple it or allow it to sustain its infection, as is the case with HIV. The only neutral mutation I can think of in a virus is one that causes it to infect a different cell type. Is this what you mean by a virus evolving to become less virulent, that it now infects a new type of cell that isn't as critical to the host organism? The way I understand it, a virus is either replicating and causing damage to the host, or it is not replicating and it is latent or eliminated. I'm not sure what the middle ground is where a virus is happily replicating yet is not virulent. I hope you understand the gist of what I'm trying to say. Anybody want to take a crack at that?
Well, it's both, Aaron. There's a tendency for the viruses to become less virulent and for the host to become more resistant. The classic demonstration of this we've talked about several times, including last week, and that's the situation with releasing myxoma virus in Australia to control the rabbits. And in the first year or two, the virus killed all the rabbits, and then over time, a new equilibrium was established where the rabbits became more resistant and the virus became less virulent. Fairly obviously, it's to the rabbit's benefit to become more resistant, and it's a little more subtle, but it's also to the virus's advantage to become a little less virulent, because you want you really don't want to kill the host if you don't have to. And I would also say that there are a lot of genes in many viruses. I would even go so far as to say most viruses that are not absolutely essential for the virus to go through its replication cycle, but rather are accessory genes that are there for the specific purpose of dealing with the immune response from the host. And here again, the myxoma virus is a great example because it's a fairly large pox virus, and these guys have dozens, up to 100 genes, I think, that are there specifically to confound the immune response of the host. And so if any of those genes is crippled, the virus can still replicate, but it becomes significantly less virulent. So there's a constant tug of war going on between the host and the virus, mostly at the level of immunity.
Right. The way to look at this is from an evolutionary perspective, where, yes, it's certainly in the virus's interest to replicate rapidly and to a relatively high titer, but it is not in the virus's interest to kill the host instantly, because then it won't transmit to a new host. And depending on how it's transmitted, it may not be in the virus's interest to cripple the host and make them, so febrile that they can't get out of bed. Because if you're an airborne virus infecting a human, let's say, you need that human to get up and go interact with other humans. So there is a selective pressure on the virus to limit its lethality and its virulence. But at the same time to replicate to a high enough level and in such a way that it can transmit to a new host.
And also, replication is not the same as being virulent. A virus can replicate very well, but not cause disease. So you have this statement here, you say a virus is either replicating and causing disease or not, and that's not the case. There are many examples of viruses replicating and not causing disease in the host, and as Alan says, killing a host is not always evolutionarily advantageous, and so many virus-host interactions have evolved so that both survive and both prosper.
There's several really good examples of this. Polyoma viruses, I always like that example. In fact, probably don't cause any disease in most people. Most people are infected with them, and they establish a persistent infection in the kidneys. And so you're carrying around these viruses undergoing a low level of replication all the time. Herpes viruses are a little more complex, but the same sort of thing, HSV type 1 that causes cold sores. You might think that only 15% of the population there's about 15% of the population that comes up with cold sores, but there's over 90% of the population that has HSV1. It spends part of its time latent and occasionally goes through a little active replication that doesn't cause you any problem, but just enough so that the virus can be transmitted.
Yeah, and I guess I should say something too then. Yellow fever virus is probably the best vaccine virus that we've had for a long time, and we got it by simply serially transferring it from animal to animal to animal until it lost the ability to kill, but it still could replicate. So that's
yeah, really.
So, Marc, what we usually do at this point in TWiV is a pick of the week. Would you like to start out?
Sure. But before we do, I'd just like to say that I think we should learn something from viruses and not necessarily destroy our ecosystem. Do your best to minimize or leave no trace. A little Cub Scout motto here: leave no trace and don't destroy your host. And our host is the Earth. So let's limit renewable resources, all that jazz. So viruses have been evolving for millions of years, and they know not to go too far. So my pick of the week is a really handy tool that I use in the lab all the time. It's great for PIs that are trying to really get a handle of lab books if they want to keep up with data, if they're analyzing data for a data analyst. It's a little app for the iPhone. It's called JotNot. And what it does is it's one of those
How do you
scanners.
spell that?
J as in Jack, O-T, N as in Nancy, O-T. So JotNot.
Okay.
I'm sure there's another one's called Scanner Pro by a company called Riedel, I think it is, but Scanner Pro or JotNot. But I use JotNot. And what you do is you just take your iPhone or your smartphone, just open the lab book, take a picture, turn the page, take another picture, turn a page, take another picture, and then it processes them, turns them into a PDF. You can move the pages around, email yourself a PDF.
Holy cow!
Now Yeah, so now with like kind of those little spy camera deals where you used to go in and, while you're dressed all in black and you snuck that night.
That's right.
Tick, 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 tick. Well, this is the fastest tool to scan. You don't want to sit there and scan your lab book. One of the problems with doing bench work is, having an analog lab book and then having your data collected on a plate reader where you've got kinetics and stuff. You sort of got to merge the two. It's really hard to figure out, then you grab the lab book and this puts the two together, makes a quick PDF of your bench notes. It's a good backup and literally instant. You take your picture, it processes it into a high contrast PDF, and then you do several. You can flip the pages around, email it to yourself and to someone else at the same time. If you're collaborating, it's the most useful tool in the lab. One of the most useful tools for me at documenting the lab books.
Cool.
Very cool.
That Dickson, if you want to use this, Dickson, you have to get an iPhone.
By the way, Mark, did you know that that's how they discovered that René Dubos was actually the first person on Earth to isolate and identify an antibiotic because of his lab book dates? It preceded Alexander Fleming by about 2 years. But that wasn't until very recently that everybody found it out. So that was cool, very cool.
Dickson, do you have a pick of the week for us?
I have a pick of the week. It was a generic pick. I read it, I believe I read this on PubMed about an outbreak in China last year of a new Bunya virus, which is transmitted by ticks, and it's got a very high mortality rate, and it's one that had never been described before. So you actually found the article for me.
Yes.
And it's got a very complicated name because it has an abbreviation which involves severe fever and thrombocytopenia associated with a novel. And thrombocytopenia means, to the other people that don't know what that is, that you lose your platelets, and that would prevent you from clotting your blood, and then you can just bleed to death, basically, from common things like brushing your teeth or something. It's an awful Bunya virus. I guess it's not a very common one because they just recently discovered it, but that's my pick of the week.
Well, that's actually something I have on the agenda for a future TWiV, so we'll discuss it in some detail. Thank you, Dickson. Rich, what do you have for us?
Good, because I'd like to do this in some detail. That's good.
Yeah.
Oh, wait, Alan, do you have to leave in 4 minutes? Alan,
I do.
Do you want to go first so that you could leave?
For it,
Yes.
Go ahead, Alan.
Sure. I'll go ahead. My totally self-promotional pick of the week this week, and I already pinged you guys with this, but it's a new blog that I just started. I'm still maintaining my regular blog, but this one is I've had these letters that my grandparents exchanged. And they date back to World War I, and they've just been sitting in a crate in my basement, and I've been moving them around from place to place. And I finally got around to starting reading them, and I realized there's a really, really cool story in these letters that I think a lot of people might be interested in. So instead of I'd been thinking I would transcribe them or scan them and just distribute them to the family. So instead, I've actually put them online. And I have scheduled the blog software so it'll be posting a new letter each day from now on. And I've already scheduled the ones a week in advance, and I'm going to do some more transcribing soon. It's called Walter and Ina. My grandfather was Walter. My grandmother was Ina. And this is, you can find it at walterandina.com. And it's what the blog tagline says. It's a story of love, war, and science. And amazing. Right now, the post that I just had come up today is the first letter that he wrote to her right after they met, and he's asking for a date. And now, this is 1924 at this point, and these are two properly raised Southerners, so there are no improprieties. But the next coming up in the next letters, which will be starting Monday, he's going to start telling his girlfriend about his work. And he is a scientific assistant with the U.S. Department of Agriculture. He's an entomologist. And he has just been, you're going to see in the letters this, he's going to start mentioning that he has been asked to go to Jacksonville, Florida. He's currently based in Dallas in 1924. He's been asked to go to Jacksonville to investigate a strange tropical disease that's been breaking out there the past couple of summers. And they wouldn't normally call the USDA for that, but they think that it may be caused by an insect. So they're calling him in as an entomologist to
cool stuff.
What's causing it. So you'll get to follow, and in looking through the letters, he describes the science as it's happening, and describes some of the false leads and the frustrations and the procedures that he's using. And you'll also see, and this ends up being clinical research, old school style.
Oh yeah.
There are no IRBs or anything like that. This is just being done.
Rich lives right in the heart of the application of that, by the way.
Okay.
To the screwworm.
Yeah, the screw fly. That's
Sure.
right. Exactly right. Yeah.
Oh yeah, the thing that he's working on at the outset is the screwworm fly.
Exactly.
And that's going to be a running theme consistently, because that still hasn't been solved in the 1920s. And but he's been called away to do this other tropical disease thing, and you're going to learn about that, and then you'll learn more about some other stuff that he does later on too.
Alan, this is fabulous.
Good stuff.
I don't really need another blog to read, but I started reading this afternoon, and it is really terrific. It's really
Thank you for doing that, Alan.
Well, it'll only be one post a day, so you don't have to commit a lot of time to it.
Alan, thank you so much for that, and thanks for joining us. I know you have to leave, so we'll say goodbye. Alan is at alandove.com and also at walterandina.com. Thanks a lot, Alan.
Thanks, Alan. Thank you. Always a pleasure.
See you, Alan.
Take it easy, Alan.
Rich, what do you have for us?
I have a pic that was sent to me by a student, Bridget, and it is a recording from the Laboratory of Applied Bioacoustics at the University in Barcelona, Barcelona Tech. There's an outfit called, let me get it here, called LIDO, Listening to the Deep Ocean Environment. This is a project of this guy at Barcelona Tech, where they're recording undersea sounds from all over the world, really, for a variety of purposes, but they caught the earthquake.
Right.
So if you go to this, you can see the earthquake, and it's pretty astounding. There's a spectrogram that goes with. I've got it going through my headphones now. It's driving me crazy. There's a spectrogram that goes along with it, and it shows the earthquake, and it's pretty amazing.
Knocked the planet off its axis. It's amazing.
Is that right?
Yeah. It slowed time down. It changed the time of the year.
Right. That's great,
bizarre.
Rich. Thank you, Rich. My pick is a light fixture from Restoration Hardware. I don't know if anyone has ever heard of this company. It's a wonderful company. And this is called, in the catalog, a steel polyhedron pendant, but in fact, it is a virus with a light bulb inside of it. You can get it in 3 different sizes. It is a icosahedral virus.
Look at it.
It is gorgeous. And the moment I saw it, it was sent to me by a former student, Eric Moss. Thank you, Eric. And I want one.
I want one too.
Don't you?
This is great. We're remodeling the kitchen, and we need a new chandelier in the breakfast room.
Oh, look at that.
My wife will go for
Go
this.
for it.
Probably not.
Probably not. Exactly right.
Well, I'll show it to her. See what she's done the whole project, right? And I've just kind of been an observer.
Oh,
I haven't made any contributions at all so far, and this is perfect.
Well, that'll do it for another TWiV, guys. As always, send us your questions and comments to twiv@twiv.tv. You can find us on iTunes at the Zune Marketplace. We also have an app at microbeworld.org where you can stream TWiV to your iPhone or Android device. Check it out at microbeworld.org. Hey, Marc, can I thank everyone, or would you like to do that?
Absolutely.
Go for it. First of all, I'd like to thank you, Marc, for having us on Futures in Biotech.
Sure, you bet. I second that one.
This was a show that terms like thrombocytopenia, squirrel monkey retrovirus, and screwworm fly were used. Thank you. It's an honor to have you on our network as well. So
it's great.
It's always a pleasure.
We get a lot of fans back and forth. I know that, and
That's true.
I really appreciate whenever we can come on. I think it was great to show the whole crew, and the crew is Dickson Despommier. Thanks you, Dickson.
Hey, Vince.
Dickson's at verticalfarm.com. And always good to see
and
you.
Trichinella.org.
And Trichinella.org. And while we're at it, medicalecology.com.
Right. There you go.
Alan Dove, of course, is at alandove.com. Rich Condit, thank you so much.
And quite welcome. This is great. And thank you. And thanks, Mark. This has been good fun.
Rich is at the University of Florida, Gainesville, home of the Gators.
How did they do, Rich? Did you follow that one at all or not?
No, I've not been following the basketball.
Okay.
I need to go to a couple of baseball games because they're doing really well.
Oh, they're good. Yep.
And I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Awesome, guys.
Hey.
I have a few thank yous as well. I'd like to thank the entire cast of This Week in Virology. It was great to have you on. I think we should make this an annual event.
Sure.
A crossing of paths.
Good idea.
And
Wonderful.
yeah, you guys have great chemistry. So thank you all coming on. It was an honor. It's certainly an honor because I'm a fan of your show too. So it's kind of like what an opportunity not only to to listen to your show, but to have you on. It's kind of, I'm touched. It's kind of, you guys do it so well.
The feeling's mutual.
You willing to come on this show? It sort of raises the bar to this show, and it says that, okay, maybe I am doing a good job on it, because I know that you guys are doing a good job and you're willing to participate.
Sure.
So it's kind of a validation as well. So it's always good. I'd also like to thank the crew that made this possible. Burke McQuinn has been on the boards, and he's had to learn all these names really quickly and keep up with the pace. He's done a fantastic job. So thanks, Burke. And also Leo Laporte for enabling this whole process and agreeing that it would be a fun idea to pull the TWiV cast on board. Lisa Kinzel, Frédéric Louis, Eileen Rivera, Tony Wang, Mike Taylor, John Salina, Jeff Stewart, Jason Howell. Thank you all for the great work you guys do at the cottage. I'd also like to thank Phil Pelletier, Will Hall for the opening and closing themes. Any comments and suggestions for FiB, you can reach me at marc@twit.tv. It's MARC at twit.tv, or on Twitter at Marc Pelletier, MARCPLETIER. For Futures in Biotech, I'm Marc Pelletier.